increase in human activity during the COVID-19 pandemic and other activities has led to the increasing need of alcohol highlighted in bioethanol. Most productions of bioethanol based on industrial methods negatively impact the environment. Various studies have shown ethanol production from fruits such as snake fruits, pineapples, or even coconuts, showing great results of ethanol production. Gaining greater results of ethanol through combining two organic waste products from using the fermentation process with different types of yeast has led into this experimental study. Hello everyone, my name is Carlos Sudeshino Krista from Elian Christian School, presenting a topic of the production of bioethanol from Salak seed waste and Ananas comosus peel waste combination. In 2017, snake fruit and pineapple are proven to be one of the most famous fruits in Indonesia. Because of its high production, snake fruit and pineapple use many unwanted wastes, such as its skin or its seed, which can be used to produce ethanol from fermentation from its high level of glucose. Therefore, combination of pineapple peel and snake fruit seeds are rich in carbohydrates, can be used as a good source of ethanol production as the subject of the study. The problem statement for this research are: first, to find the effectiveness of ethanol production through the combination of snake fruit seed and pineapple peel. Second, to find the amount of ethanol production percentage from two types of yeast, bread yeast and tapa yeast. And third, to find the type of yeast and the best concentration of yeast and time fermented to get the maximum amount of ethanol production. Snake fruit seeds and pineapple peels have the highest carbohydrates within its fruit parts. 28.98% of cellulose and 59.37% of hemicellulose for snake fruit seeds, and 14% of cellulose and 20.2% of hemicellulose from pineapple peels. Fermentation occurs by adding yeast, which converts sugar into ethanol and carbon dioxide. Two types of yeast are used in this experiment: bread yeast, which can be used to produce ethanol as a concentration of 18% of the fermentation broth, and tapa yeast, which produces ethanol with a maximum amount of 20%. Combination of snake fruit seeds and pineapple peel increases the amount of ethanol production due to its combination of high carbohydrates. 1 kilogram of each fruit and a refractometer is also prepared for testing the percentage of ethanol prepared for this experiment. Samples are manually cleaned to remove excess debris and then grinded for pineapple peel and cut for the snake fruit seeds into small pieces. Each fruit samples are divided into 6 beakers of 150 grams. Different concentrations of bread yeast and tapa yeast of 3.15 gram, 2.25 gram, and 1.35 grams are prepared, each dissolved in 100 milliliter of water. 6 mixtures of snake fruit seeds and pineapple of ratio 1:1 of 300 gram peels and seeds are prepared in 6 gallons, added to 100 milliliter of dissolved yeast bread and yeast tape, with different doses of 200 milliliter of water added in its labeled gallons. The mixture is then allowed to stand for 4 until 6 days. The process of fermentation works by allowing carbon dioxide to be released and exit through the gallon pipe connected to its liquid. Samples of the fruits are then measured using a refractometer to determine the ethanol content in its units or percentage. The drop-off samples of bioethanol are then put in the prison assembly in the refractometer and then looked at the IPs for day 4, day 5, and day 6. Results shows that the highest percentage of ethanol produced is at 10%, produced from 3.15 grams of bread yeast and 2.25 grams of tapa yeast. It shows that the best yeast used is tapa yeast of concentration 2.25 grams, since it ferments the fastest yet the highest percentage in just 5 days. This experiment proves that the highest amount of ethanol is made from tapa yeast of 2.25 grams as the best fermenter due to its fastest amount of time to ferment. Snake fruit seeds and pineapple peel combination is very rare to be used. Snake fruit seed is rare since it's harder to handle the fruit sample. Therefore, this can be used as an alternative of study of ethanol production. The future work for this experiment is to find the best method to harvest best ethanol and produce a useful product using ethanol for medical and educational purposes. Remember, waste is just waste if we waste it. Thank you and God bless you.